presenter
I
mode
see.
on Keynote,
I see.
I don't see anyone else.
Okay, perfect.
So
thank you very much for agreeing on this. We are looking forward to your talk on your new preprint in BioRxiv.
Excellent. Thank you. It's a pleasure. Wonderful. So you wanted to just go ahead and get started now?
Yeah. Maybe you introduce yourself a bit first and then go into your talk.
Certainly. I'm Peter Kasson. I'm at Uppsala University in the Department of Cell and Molecular Biology, and also at the University of Virginia. And I'm going to talk about the recent preprint we have on triggering and chemical control of single virus fusion within endosomes. This is work with Saurav Halder, who is pictured on the lower right, as well as Kenta Okamoto, both when they were in my laboratory, and Saurav is continuing in my lab. So we're broadly interested in pathogen membrane interactions, and in particular in infection by influenza and emerging viruses. And influenza is a prototypical envelope virus in that it infects cells via the endocytic pathway, as do most, although not all, envelope viruses. Influenza infects cells in mid-late endosomes, and so it binds to sialic acid terminated glycans on the cell surface, is endocytosed, and then acidification of the endosome triggers a conformational change in the hemagglutinin coat protein that causes insertion of small fusion peptides into the target membrane, refolding of the hemagglutinin coat protein ectodomain, and ultimately fusion. And this fusion results both from the refolding event and from complex interactions of the membrane inserted portions of the hemagglutinin protein. And so a lot of the other work that we do is on the details of these protein membrane interactions and how they result in fusion. But one fundamental challenge is how do we track and measure these fusion events in as precisely controllable, because we are interested in the membrane physical chemistry of this process, but as physiologically relevant a system as possible. And there's been a fair amount of beautiful work done on tracking single endocytosis and fusion events of influenza in live cells. I show on the left a figure from Xiaowei Zhang's work, where they tracked individual fluorescently labeled virions from the, you can see it, just sort of a slight outline from the plasma membrane to, in this case, a fusion event that occurred in the endosome, relatively close to the nucleus. And then Greg Malikian has also done work shown on the right, looking at fluorescence dequenching traces, again, of individual labeled influenza virions inside live cells. So that provides a, at least at a cellular level, a physiologically relevant context for measuring fusion events and for tracking fusion inside the cells. However, this lacks the temporal control or the chemical manipulability of synthetic systems. And so shown on the right here is some work we did actually with Zika virus, although we've done the same for influenza, where we bind viruses to a synthetic membrane, and I'll tell you a little bit more about how that works in a slide or two, and then can trigger fusion in a much more precise fashion. We can control many of the elements of this, and in this case, we could look at the way that the fusion kinetics vary with pH over tens to hundreds of fusion events that are all precisely temporally aligned as to when the pH trigger occurred. And so doing that in a live cell system, both the temporal control, the temporal alignment, and the chemical manipulability is extremely challenging. And so the work that I am talking about today is an attempt to bridge these two to some degree, to permit fusion, measurement of fusion to endosomal membranes, single event tracking, but at the same time, precise temporal triggering and chemical manipulability. And so the underlying system that we and others have worked on previously is a measurement of fusion of single viruses to synthetic membranes. So this was for influenza, first reported by work from Dan Floyd and Antoine Van Nooyen and Steve Harrison, and also from Keith Wenninger's group, where in a system much like what I'm describing here, we can construct synthetic target membranes inside a microfluidic device, allow viruses to bind to target either synthetic bilayers or in our case liposomes, trigger fusion by a pH drop, exchanging the buffer within the microfluidic cell can be done with fairly high temporal precision, and then measuring the single lipid mixing or fusion kinetics by single viruses. And so you get a movie like what is shown on the left here, where each of those spots is a single influenza virion, and we can do a time trace of the fluorescence intensity of these viruses, and there's a jump when a Texas red dye that's in the viral envelope dequenches upon lipid mixing, that there's an increase in intensity of that spot. And we can measure the waiting time from when the pH drops in the flow cell, which we can also measure precisely via a pH indicator, separately introduced, to an individual dequenching event. We can then compile these as a cumulative distribution function, as shown on the lower right, to get a kinetic profile of individual events, and query this in a number of different ways to understand the kinetics and mechanism of fusion. And being able to do this from single events is valuable as opposed to bulk kinetics, because the shape of this distribution contains a lot more information than just the mean value. So fusion to the inside of the endosomes, the work I'll tell you about today is the sort of motivating idea is to be able to infect cells with influenza, arrest endosomal acidification, isolate endosomes with trapped virus. So remove those from the cell and then immobilize those in a flow cell, similar to what I showed you, retrigger fusion and measure individual fusion events. So this offers the potential to maintain the physiological membrane environment for fusion, but at the same time introduce the ability for chemical triggering and control that is largely missing from the live cell experiments. So influenza fuses in mid-late endosomes. This is a schema from Judy White's work, that it can fuse either in Rab5, Rab7 double positive endosomes or Rab7 single positive endosomes. It's somewhat later than most flaviviruses, so later than a virus like Zika or dengue, and at the same time earlier than Ebola and SARS. Ebola enters in NPC1 positive endolysosomes. And the common marker, if you look at the schema, the common positive marker that's relatively unique to the influenza entry compartments is mannose-6-phosphate receptor, M6PR. And so that while the endocytic pathway is a continuum and contains a lot of complexity, and one would not say that a single marker can perfectly identify any given compartment, that M6PR shows relatively good correlation with influenza fusion compartments. So what we've done is to infect endothelial cells, in this case, this is BHK cells, with influenza virus, arresting the endosomal acidification with ammonium chloride, although this can also be done with other agents, and then mechanically lyse these cells and sucrose density gradient purify the endosomes. And so shown on the left is an example density gradient. This is a step gradient, and you can see distinct bands with different membrane fractions. And if we take fractions through this sucrose density gradient, one can see immunostaining for hemagglutinin, and also probably doesn't render quite as well, but immunostaining for mannose-6-phosphate receptor really coming out in fractions 5, 6, and 7 here, which correspond roughly to this late endosomal band shown on the left. And so these are our diagnostics that in these fractions, we are obtaining both hemagglutinin positive and mannose-6-phosphate positive membrane particles. If we do cryo-EM on the purified vesicles from these fractions, we see a lot of endosomes, as shown on the bottom here. They vary in size. Some of them are multilamellar. And we also see a relatively small number, as we'd expect, of viral particles or apparent viral particles labeled with a V within some of these endosomes. The putative viral particles are identifiable by a density pattern that is similar to that we see for hemagglutinin spikes en face in isolated virus on cryo-EM. And it's interesting that these viral particles are, at least in the few we've observed, are often in somewhat larger endosomal compartments. As you see, there's a fair amount of pleomorphy here, which is again what one would expect for such for endosomes. So if we then take these endosomes and we immobilize them inside a flow cell, and I should say before infection, we Texas red labeled the influenza virus, and so we can see a relatively sparse pattern of Texas red positive spots, which is what we're looking for because we infect at MOI no greater than 1, multiplicity of infection, so the average number of infectious particles per cell, because while we want to capture influenza virus conjugates at the same time, loading a given cell with a very large number of influenza virions can perturb the endocytic physiology, and we're trying not to do that. But in any case, if we look at the red staining particles, if we immunostain with mannose-6-phosphate receptor antibody, and then examine the colocalization, we have a relatively large number of M6PR positive spots, and that our Texas red positive particles show relatively good colocalization with the M6PR positive spots. The other control that I'm showing you here in the bottom half of the panel is how we can do pH control for influenza virions that are now inside an endosome as opposed to directly exposed to external buffer. And so if we, when we do the buffer exchange, if we include an ionophore, in this case FCCP, we get fairly strong pH control of the lumen. And so this is shown here. If we take carboxyfluorescein inside vesicles, carboxyfluorescein is pH sensitive. And if we then do this pH, this buffer exchange with FCCP in it, that the fluorescence intensity traces from these two spots here show a bit of a bleaching pattern for carboxyfluorescein, which is what we expect, but a sharp and relatively well synchronized drop in intensity corresponding to the pH change inside the vesicle lumen at the time when we flow the FCCP containing buffer into the flow cell. So it looks like we have M6PR positive endosomes containing Texas red labeled virus. We can control the pH. What then happens when we do this and trigger and monitor lipid mixing? So this is shown in a single trace on the left here, and then a bunch of slightly less pretty, but very real data on the right to show that this isn't just a single, beautiful and completely unrepresentative trace, that we get a number of different Texas red labeled particles show dequenching events, although certainly not all of them, and that they show relatively sharp dequenching that is similar to what we see to the fusion of virus to supported bilayers or to the outside of liposomes. And so we are indeed detecting single virus endosome fluorescence dequenching events in a pH-triggered manner, as we would expect. If we then take a large number of these and compile a cumulative distribution function and compare this to virus from the same prep fusing to the outside of 100 nanometer liposomes, we see some similarities and some differences. That overall, the fusion to endosomes has this slight delay in the CDF at longer time points. If we scale these, though, in the inset, you see that the fast phase of fusion to endosomes overlays almost perfectly with fusion to the outside of liposomes. So it looks like there are these two populations kinetically of viruses inside the endosome. One that seems to be fusing with kinetics exactly identical to fusion to the outside of liposomes, and a second, smaller population that appears to be fusing somewhat more slowly. I should also add that to exclude any virus that might have escaped the endosome during mechanical lysis and purification, we also, in acquiring these single virus traces, we, rather than labeling with M6PR in the antibody in these actual production experiments, we label with an anti-hemagglutinin antibody, and any Texas red positive particles that also stain with anti-hemagglutinin are solvent exposed and therefore not fully encapsulated within endosomes and are excluded from the analysis. So this is truly fusion from within endosomes, and it again is quite interesting just how similar most of these fusion events are kinetically to fusion, or at least lipid mixing to the outside of liposomes. And so this was quite an interesting result to us. both proving that this can be done and that most of it is kinetically similar to fusion with pretty different curvature. This is consistent with some results that we published earlier this year, where we examined influenza viral lipid mixing to the exterior of liposomes with different radii of curvature, or even to a planar supported bilayer. And there, the cumulative distribution functions essentially overlay independent of the target membrane curvature. So this is consistent within the idea that if we reverse the curvature and look at fusion to the inside of endosomes, that might not have a huge effect on lipid mixing rates. Also, in a brief ad, I'll refer extremely quickly to a ChemArchive paper that we published recently, where we used silicon nanoparticles to manipulate membrane deformability. So if we use different radii nanoparticles, this changes the membrane deformability because it affects the relative magnitude of spontaneous shape fluctuations of the membrane. And we can then bind influenza virus to GD1A glycosphingolipids on the surface of these supported bilayers, and similarly perform single virus lipid mixing experiments. Here we saw that in a nanoparticle radius dependent manner, we were able to observe slowing of lipid mixing kinetics compared to empty liposomes. And we also did a more extensive analysis that I'm not going to go into today, where it looks like the differences in deformability affect apparent fusion protein stoichiometry. But the take home for today is that it appears that altered membrane curvature probably does not substantially affect lipid mixing kinetics, but altered membrane deformability does affect lipid mixing kinetics. So some of the outstanding questions at this point in this work are whether rates of fusion pore opening are also comparable between influenza fusion to endosomes and to the outside of liposomes, or whether they show the similar independence as lipid mixing. And so that's one question that we're really interested in. Another is what distinguishes these kinetically different virus endosome populations? Can we start to tease apart what might control whether a virus would have that faster or that slower fusion profile? Also, given that about 7% of the viruses or Texas red positive particles can have fusion successfully triggered, we're interested in what controls whether a virus inside the endosome is fusion competent. So obviously not all of those Texas red positive particles might be bona fide virus. There have also been estimates by a number of other scientists that a relatively small fraction of endocytosed influenza virions are actually infection competent. A larger number of those might be fusion competent. So the 7% number is not crazy low, but whether we can use this sort of analysis to get a better sense of what controls infectiousness at the late endosome stage, or at least fusion and potentially uncoding competence. So those are some of the obvious next questions in this work. Some of the other applications that this should be useful for, that we're really excited about, are obviously looking at cellular restriction factors, at looking at perturbations to the lipid environment either exogenous perturbations, now that we can remove the endosomes and they're isolated in a flow cell. That means that a lot of the methods of lipid manipulation, such as sterol exchange that we and other people have previously done, are accessible, as well as pharmacological manipulations to the cellular lipid environment. Also using this platform to examine the fusion of other viruses within endosomes is another key application. So to briefly summarize, individual fusion within endosomes can be controlled and observed outside of cells in this isolated virus endosome system. This control enables kinetic analysis of how factors such as composition and asymmetry, which are quite different between the endosome and synthetic liposomes, as well as curvature, control fusion kinetics. And perhaps surprisingly, most of the influenza endosome conjugates we've studied show lipid mixing kinetics similar to that between influenza virus and the outside of liposomes. And this is in agreement with our prior data suggesting that curvature does not strongly affect lipid mixing kinetics, and our other recent data suggesting that membrane deformability does affect lipid mixing kinetics. So with that, I'd like to thank the people who did the work. This was largely work by Saurav Halder. Kenta Okamoto did a lot of the initial system development. The work on insensitivity to liposome size was done by Bob Rowell, who's now at Williams, and the work on deformability was done by Ana Villamil Giraldo. And that's another wonderful story that I'd love to talk with people about later. And if people are either interested in joining the group or have questions, if they want to contact me either via email or on Twitter, that would be great. Thank you.
Thank you very much, Peter. That was fantastic. Thanks a lot. So we'll have a couple of questions. I think I want to start with my question first. So the curvature, this is probably very, very interesting for me to see that in the assays that you're doing with endosomes and liposomes, curvature doesn't play a big role in fusion. So does this mean, for instance, is there first of a dynamic range that we can play with, or if we go to SLBs, it would be the same?
So, oops. At least with one SLB system, this was a sort of a very simple glass-supported bilayer, if I recall correctly. We saw the same. Those are the blue dots here. There are obviously a number of different SLB systems, and I think for fusion, for a fusion pore opening, there may be more differences, especially with SLBs.
Okay, so there are a couple of questions from the audience. So first, you answered a couple of them already in the talk. Ilya Leventhal asked, do these cumulative distributions imply all the particles are fusion competent? You even gave a number, right? They are not all fusion competent, it's 7%. And Pablo commented that for HIVs, it's also not all of them fusion competent. And Pablo says, do you think you could arrest lipid mixing, the hemifusion, by tuning the lipid composition?
Quite possibly, and people have done a lot of work with things like lysolipids to tune the lipid composition. I think one of the challenges from, it depends on whether this is a interventional or a mechanistic tool. I think one of the challenges with tuning the lipid composition is that obviously changes a number of things at the same time. It changes some of the bulk materials properties. It potentially changes spatial organization of the membrane. It also changes some of the properties of some of the highly curved fusion intermediates, and maybe some of the sort of free energy barriers to different lipidic steps in fusion that depend on individual lipid molecule properties. I'm not sure anyone has done a full arrest, but certainly people have altered it. That's part of why we went into using silica supports as a way to try to tune this by altering mechanical properties without changing lipid composition.
So related to this, when you have the liposome system, you always use GD1A, no other glycolipid, right?
Yes.
Is there any other glycolipid that would show the same effect?
So flu combined to a number of different glycolipids as well as a number of different glycosylated proteins. And so there's some back and forth as to which glycosylated lipids and proteins are the physiologic receptors on the cell surface for binding. Also, the lipid binding affinity varies between for different glycans varies between strains of influenza virus. So one could definitely use other glycolipids for, especially for other strains. This is X31 that binds GD1A well, but some of the other viruses don't bind GD1A so well. And I think there's been some work recently by some of my collaborators looking at other glycans to try to engineer this.
Okay, Pablo also asked, did you test fusion blocking antiviral compounds?
We have not yet. That's something that we're very interested in. And obviously the endosome system gives a way to look at things that block fusion, beyond what can be, just the way that one can test inhibitors in the semi-synthetic system. So the semi-synthetic system is a pretty straightforward way to look at that. But the endosome system gives us additional methods. We've done a lot of work, not here, but presented at this year's Biophysical Society on antibodies and blockade of fusion, but we haven't looked at small molecules yet.
Okay, there is one question from Zhang Shi. Can you explain more on the membrane deformability part? Is this a result of different size particle inhibiting different fluctuation modes of the membrane?
We haven't looked at the precise fluctuation spectra of the membranes. That would be an interesting question. I'd have to think about the measurement methodologies for that. But sort of intuitively, if one, by fluctuation dissipation, that the free energy of an induced deformation mode of a membrane should be the same as the free energy of a membrane spontaneously acquiring the same shape through spontaneous fluctuation, and the the silicon nanoparticles greatly reduce the shape heterogeneity, certainly static, and also we believe, and from what we can see on cryo-EM, at least the if one assumes ergodicity, the distributional shape heterogeneity. But we haven't sat there and measured the fluctuations. That would be a really cool experiment.
Related to this, there's another question from Raya Sorkin. Says, is the bending rigidity important and thus can be maybe tuned by lipid composition, or maybe tension changes for your supportive membranes and then affects the fusion?
So let's see, I'm not sure I understand the tension part of the question, but the bending rigidity is almost certainly important. Someone can have to think about whether the tension, the baseline tension, I think, should not be different for the supported membranes versus certainly a planar bilayer. I don't think there are obviously a lot of people who think more about tension than I do. That the induced tension for some deformation I think that is more or less the same as the bending rigid, as the deformation free energy, right? Because again, tension is really a delta G property, and so it's a question of delta G this way, delta G that way. I think those are really linked when you're thinking about bending a membrane. But it's a good question.
Okay, thank you very much, Peter. That was amazing. Thanks for your time. People enjoyed it very much. There were a lot of compliments that I didn't pass on you. So thanks a lot.
Thank you. It was my pleasure. And if people have further thoughts that they'd like to share individually, I'd be particularly interested, especially in this relationship between deformability and tension. So thank you. I appreciate the opportunity to share our work.
Thank you very much.